For today we're going to look at genetic disorders, and we're going to look at one particular one called polydactyly. Now before we do that, there's a couple of definitions that I want you to know and understand, and the first one is the word gene. Now a gene is something that controls your features. You may have seen a previous video about the specific details of what a gene is, but for now, for the purpose of this video, I want you to understand it as something that controls the kinds of features that you have. Now genes have related to them what we call alleles. Now alleles are versions of the gene, so one allele is one version of the gene. Now to just give you a better idea of that, let's have a look at a couple of examples. One characteristic is called tongue rolling. That's whether you can roll your tongue or make a tube shape with it, and the gene, as I've highlighted in blue here, is the tongue rolling gene, so that decides whether you can roll your tongue or not. And the two alleles that we have are can tongue roll and cannot tongue roll. So the alleles are can and cannot, but the gene, the overall thing, that the overall characteristic that we're talking about, is tongue rolling. Another example would be your ear shape. There are two types, rounded and attached, or sometimes attached and unattached they're called, but either way the gene is ear shape, and the two versions are rounded and attached, as I've written it down here. You can also look at it in plants. So a gene for in pea plants, in humans and animals, height is a bit more complicated than just having tall and short, but certainly for peas the height is the gene, so peas have a gene for height. The two alleles are tall and short. So all these in green are the alleles, the versions of the genes, and all these in blue are the actual genes. They describe or control the features. Now going back then to the topic, which is a genetic order called polydactyly. Polydactyly is a disorder whereby you may have extra digits, for example an extra thumb or an extra finger on either of your hands, or it can also end up with extra toes. Not just one, but you can have sometimes 3 or 4 extra toes on the feet or extra fingers on the hand. And this is a genetic feature that some people have, and we're going to look at how this can possibly be inherited. So let's have a look at an example here. We've got two parents, a male and a female. The male in this case is affected and the female is unaffected. The affected male, that means, has polydactyly. Now in order to describe the genes and the alleles, the gene is for polydactyly, but the two alleles are affected and unaffected, and I'm going to give a little code for each one. So a capital P we're going to say is affected, and the small p we're going to say is unaffected. So which two alleles will the father have? The father does have two alleles. He has one from his father and one from his mother, and for this example we're going to say one from his father, one from his mother, each one a capital to say that he is affected. For the female we're going to have two which are alleles that show unaffected. Now in terms of the baby that they have, will the baby have polydactyly or will the baby not? Well, it depends on how they inherit these alleles, and I've done these as capital letter and a small letter for a particular reason, because the allele for being affected by polydactyly is what we call dominant, and for unaffected is recessive. Okay, now recessive, the recessive allele means it will only show up if the dominant one is not there. If the dominant allele is there, then the dominant allele will show up. So in terms of polydactyly, it is a dominant genetic disorder, so that means if the capital P, the dominant allele, is there, then the person is going to be affected. But how do we know in terms of the children whether they're affected or not? Well, when the father passes on these alleles, he can only pass on one or the other. He can't pass on both, because remember, during meiosis we halve the number of chromosomes. We go from a double set to a single set. So what will happen is the father will pass on either one of these. Now in this case it's fairly simple because it's both capital P, and the mother similarly in her eggs will pass on one or the other. So what you'll end up with is sperm cells, along with all the other thousands of alleles that have been carried, the ones we're looking at, will carry capital P or capital P, and the mother will carry either small p or small p. She can't pass on any others in the eggs because that's the two that she has. Now, it's a question of looking at what the chances are of getting each pair of alleles in the baby. So imagine, let's give these numbers: sperm cell 1, 2, egg cell 3, 4. We could have sperm cell number 1 meeting egg cell 3, so the two alleles there would be capital P and small p. You have to be when you do this in the exam, you have to be very careful to be clear what's capital and what's small or lowercase. P wasn't probably the best one to choose, but either way, I'm being very careful that you can see the difference. So we've got one sperm cell here could join with this egg cell. This sperm cell number 1 could join with number 4. So again, you would have capital P, small p. Sperm cell number 2, capital P, can join with number 3. So we have small p there. And the fourth possibility is the capital P, sperm cell number 2, joining with egg cell number 4. So again, we have capital P, small p. Now, these are the alleles, the possible alleles that can join together to make your baby. Now, we said that the dominant allele, if it's present, it will show up. So in this case, we're going to have an affected baby. This one is affected, this one is affected, and this one is affected. They are all actually going to be affected because they all have a dominant version of the gene. Okay, so you should be able to do a cross like this to show how the alleles can combine to produce the offspring. Now let's do a different version in a different way and take a different example. So let's imagine we've got two parents. Let's say one parent has these alleles and the other parent, for argument's sake, has the same alleles. So we can say they're both affected because we know that they both have a dominant version of the gene, and we can go through these steps to find out what the chances are of the babies being affected. But rather than doing it this way, we're going to do it a slightly different way. We're going to use what we call a Punnett square. We've come across this once before. You need a noughts and crosses grid, and you put the father's alleles on the top. You could put the mother's if you want, but it doesn't matter. They're both the same in this case. But the father can only pass on a capital P or a small p in the sperm cell, and the mother, let's put just here, father, and the mother again can pass on a capital P or a small p, put mother along the bottom. Okay, so what are the possibilities? Well, these two can combine, two capital P's. These two can combine, capital P and a small p and a capital P, small p, small p. Okay, so you can see on this last one here, it's starting to get slightly confusing because I'm not drawing it very clearly, but these are two small p's. That means out of these children, all the possible children here, we have this one is affected, this one is affected, this one is affected, this one is not affected. So in terms of how many affected, we've got 3 affected to 1 unaffected. Just squeeze that in there. So that's the possibilities of the child. The ratio is 3 to 1, or 75% chance of being affected, or 0.75% chance of being affected. So you can write it in these ways. This is 0.25 or 25%. Okay, so you should be able to do a genetic cross. This is what we call a genetic cross for this particular genetic disorder called polydactyly. You must remember that it is a dominant genetic disorder, so the dominant allele will cause a person to be affected, and these are the possibilities in terms of the crosses. You might get a cross that you have to do for yourself, for example something like this, or whichever way around, but you should be able to follow these steps in order to show the genetic pattern for this particular disease.